So this session is about exploring and visualizing multiplexed molecular profiling datasets. And this morning, there's a session on multiplexed molecular profiling data generation tools. And so I was at one session, and it's about imaging-based multiplexed data generation tools. And another session is about sequencing-based tools. And so this afternoon, so we're going to talk about data analysis tools. So this session is about visualization and exploration of the data sets. And so with that, I want to perhaps introduce, myself. So I'm Yong Yao, actually managing Brain Initiative Cell Sensors Network, a program officer at the National Institute of Mental Health. Can we have Ananda? Could you?
Yeah, so this is Ananda Roy from NIH Common Fund. I'm involved with HubMap along with 7 other Common Fund programs.
Mike Pacing?
I'm a program director at NIH-HCA. I work in functional genomics. I work on things like Common Fund 4D nucleome, ENCODE, unsolicited awards.
Thank you. Fiona?
Hi, everybody. This is Fiona Ginty. I'm based at GE Research Center in upstate New York. Been involved with multiplexing and HubMap for the last year, and also have a couple of other oral ones where we're looking at colon cancer heterogeneity and immune response in ductal carcinoma in situ. And our group has developed protein multiplexing methods over the last 10 years and published on those and sharing those methods out with the community. And so just very interested and happy to take part in this group.
Thank you. Ilan?
Hello, my name is Ilan. I'm a software engineer, developer in Niels Gellenborg's lab at Harvard, working on HubMap, and we are actually building a viewer for web-based viewer for the high bit depth imagery that multiplexing generates. So it'll be hopefully in HubMap and maybe in other projects soon. Yeah.
Thank you. Benedict? Benedict Anchong?
Yes, good afternoon. I am a PI in NIHS, and I'm working on developing methods and for single cell analysis, particularly with focus in toxicology and cancer.
Thank you. Candice Lay? You are muted. Perhaps you are muted. Okay, Jennifer Root.
I am unmuting myself. I'm Jenny Rude. I'm at the Broad Institute in Aviv Regev's group, and I'm just going to observe. I'm not actively participating, but I'm interested in what you all have to say.
Thank you. John Hartman.
Hi, this is John Hartman. I'm with the University of Michigan, the Michigan Kidney Translational Medicine Corps, and I'm observing also.
Mark Keller.
Hi, I'm also at the Harvard Medical School Gellenborg lab working on tools for visualization of spatial data. And right now I'm also just observing.
Sabrina.
Hi, everybody. Nice to talk to you today. I'm from the Medical University of Vienna, and I started now postdoc position there, and I started a new project. And I'm mostly observing, but maybe I'll join the discussion if I can. That would be great.
Okay, thank you. Philip?
Hi, Philip Oberdorf. I'm with the NCI Human Tumor Atlas Network on the program side, observing for the most part.
Yeah, so for today's session, we need a scriber. So Philip, could you do that? Just as you did this morning?
I did it so well, right?
Exactly. Yeah. So, so if you can open the doc, so you will see the discussion topics listed in today's document. And briefly, I just probably just go through several bullet points. And the first is what tools can be developed to capitalize on the new features of multiplex profiling platforms. Second, where do we find multiplex data sets? For example, a tissue which has both single cell gene expression and spatial profiling data. Third, what types of information should these contain to be useful to others? Fourth, where do you find tools to explore and visualize such data and outputs from pipelines? Fifth, what are the current open challenges for exploring and processing such data sets? And do we have any other discussion topics that you want to add?
I can't seem to get to the document, which.
So that's in KI Storm website. And so, you know, before you join the Zoom meeting, you have blue button next to green button, open doc.
Yeah, so which number are we on now?
That's 8E.
8E, okay.
Yeah, 8th session.
I pasted that in the chat.
Yeah, so so the multiplex the methods are the multimodal. That I think this morning actually we're mostly focusing on multimodal molecular profiling, and so instead of just doing one modality at one time, and so simultaneously detection of DNA, RNA, protein, or states, even combined with the physiology. And so now becoming more and more popular, and quite a few methods have been reported. And so what kind of computational tools need to be developed and to visualize and to explore this kind of data set, and what unique features that may be missed in the individual single modality assay, but can be captured through simultaneous multiplexed molecular study. So that's something to be discussed today. So maybe we can talk about first several protocols. Maybe some of you are familiar which protocols you are using.
Maybe can I just throw in something there, just because we had a similar discussion this morning. We didn't get quite as far as the analysis, but at least from a problem statement perspective, and I'm coming at this as a biologist, not as a computational person, but working with closely with computational folks on single cell profiling and data integration. But one of the challenges we have is that more seamless movement from the generation of single cell data to the final data set, and then matching them back to the original data set. So sort of an interactive view between the single cell data, the spatial information, and the original images. And as of today, I mean, we have our own in-house homegrown methods there that we've developed in R and so forth. But I think, you know, that really has to be a priority to, you know, for the field.
So you're,
I don't know if anyone else has a...
so you're talking about imaging data?
Yes, I presume that's what we're talking about here, right? Or are we not? If we're not, then I guess I'm in the wrong group.
I think this is, yeah, imaging data is one type. I think there are other multiple sequencing based.
There is a
Sure.
there is a dedicated session. There's a dedicated session on imaging, and I think 9A or B that's led by Dennis Shapiro.
Okay.
Yeah.
Thank you. It's okay. I understand.
Apologies for that.
Yeah.
Maybe what I
I
maybe
think multiplex here. So so for example, in my program, there are some people are doing actually patch seq, so which basically record transcriptome sequencing and electrophysiology recording, and also imaging morphology. So 3 different modalities from the same.
okay.
So, you know, there are different ways of doing experiments. I think that there are probably other protocols as well to be discussed this afternoon. Anybody can share or want to share?
But I think, this is Benedict, I think really integrating has to be within the context of across technologies or just the same measurements from the same sample. When you, in terms of visualization. So I think these are 2 different approaches that I think that actually requires different challenging computational methods, I guess.
So what kind of methods are you using?
So I think currently most of the methods we do is that we focus on one, most of the time we try to look at data reduction methods, isn't it? But in terms of sequence, RNA-seq, for example, or omics. But of course, I think we need methods that can really help in the integration of data across, not just within like methods that data sets that come from the same sample, or data sets that come from different samples, or data sets that come from different scales. So I think, I don't know if there are methods out there that can handle this kind of problems.
They want to jump in? So, yeah, so I'm not not very clear what particular method. Maybe this is some challenge that we should note down how to integrate between different modalities. Yeah,
so, well, maybe what I was driving at was that I think before we talk about integration, we need to be able to clearly define like what is feasible and in terms of the data generation process, isn't it? Because, and what are the ultimate goals for the integration? That is what I'm just saying, that methods have to be what context. So I'm just putting it out there. If we have methods that can really generalize to different contexts or just specific or with respect to a particular context.
So for modality, so like sequencing-based approaches, like RNA-seq or ATAC-seq. So both are, you know, a lot of methods to analyze each, but combine them. What kind of advantage with the new analytic tools generate additional information. So is that clear or not clear? I don't think there's a very good comparative study so far, right?
Yeah, I see. So we are focusing mainly on just, for example, RNA-seq. Is that
no RNA-seq? Yeah, so RNA-seq or ATAC-seq, these are probably most used right now. And then there also can be like combination of DNA methylation with high C, and that also very recently published method. And so all these simultaneous assay, what advantage they are in comparison with analyzing individual molecule species separately, respective analysis. So that's not very clear at the moment.
Okay, I see, okay.
Right, so I hope this kind of challenge, I mean, this is something to be discussed. How do we explore this kind of data?
In this session, are we, or is this session dedicated, at least in part, to try and figure out how to integrate RNA sequencing with spatial imaging at all, or single-cell RNA-seq in the context of CISIF or other spatial proteomic analyses, or is that discussed somewhere else?
Yeah, so, well, this is some people are trying to image a protein and RNA from the same cell. But that kind of technology, to my knowledge, is not very well developed yet.
I guess I was more thinking combining a single-cell RNA-seq data set with multiplex proteomic imaging data set from the same sample, not necessarily in the same experiment.
So this is so data integration. Okay. So yeah, so
if that's not the group for it, that's fine. I just thought that falls under visualizing multiplex data sets, but.
Yeah, that's what I was thinking as well. I was thinking about generalizing, like visualization of different kinds of data sets.
Okay.
So that's why I was thinking that, is this group just kind of generalizing the method, or just focusing on a particular technology and the data sets, or like visualizing across modalities?
Well, so I think probably we can expand. So instead of focusing on simultaneous recording, but we can also expand to more general question, how to integrate across modalities. Is there something I think we should tackle here, right?
Well, maybe one thing we did in our session this morning was define, at least maybe you guys already know each other, but what types of data sets are you already working on? At least sort of just to get a grounding on what kind of problems you're facing. And then from there, how do we define, you know, later define the solutions? But, you know, would that be helpful to start at that sort of level?
Okay, so maybe I can probably start first. If anybody have something to say, share, please just jump in. So my program is about the cell census of the brain tissues. And so far, for the molecular profiling technology used include single-cell RNA-seq, ATAC-seq, DNA methylation, merfish or seq-fish, and then anatomical morphology and imaging, and you know, in the future, probably also calcium imaging, this kind of a physiological recording as well. And so the data are generated and how to integrate them together. And so there are some joint analysis group, for example, for the molecular data sets to integrate ATAC-seq, DNA methylation, and RNA-seq data. So these three modalities together. And so when actually the paper just published in bioRxiv a couple of weeks ago, reported from this Brain Initiative consortium. So the group, so basically they are using a gene body information to map chromatin, RNA, and DNA methylation data together. And so these are from individual brain samples, cell samples, not simultaneous recording, but recorded differently from just post-experimental joint analysis. So it's it's a cell ensemble type of analysis. So is that something that we should talk about?
So this is Anand. I was just going to jump in and say just a couple of things. One is that it's my understanding that what we are trying to achieve here in this session is, as you have outlined quite nicely, that really what are the questions, what are the challenges that we face, and then perhaps use the next session for 75 minutes to answer some of those challenges. And some of the challenges I think we have started discussing, but we need to articulate better, that when you are talking about ATAC-seq versus RNA-seq, it's somewhat not that difficult. How Satija has done, many other people have done that. These are going to give us some sort of a similar output, in the sense that they will both lead to definition of perhaps new cell types, and it's somewhat easier to compare those two data sets. But when we are talking about two different scales, for instance, how to integrate imaging data with omics data, it's a whole new beast. And do we have really any way of doing that? And how can we then combine RNA-seq data, transcriptomic data with proteomic data or metabolomic data? What are the challenges there? How do we begin to even think about combining those for a better visualization?
So I think I agree with what Fiona said earlier. For this session, I think it makes sense to go through those that actually produce or analyze data and what those data types are, right? To have the most productive discussion, as opposed to, theoretically addressing data where we have no representation in this group. Does that make sense? Do we have people that are working on single cell ATAC-seq in this group? And do we have people that do, obviously most people will probably have experience with single cell RNA-seq, but do we have people that do DNA methylation?
I mean, I work with collaborators with people who can generate that kind of data. Mm-hmm. But I mostly work with proteomic. So I suspect we might have a diverse people, as I said, because typically most labs, they tend to focus on one technology, and you would hardly get a big lab that has all the technologies in-house. So what I suspect is that what is out there is that we have data sets, even for the same biological system, but they might come from different technologies. And I think that is a challenge. Like, how do we, I think that's a big problem. Like, how do we use all of this information to study the same process?
Mm-hmm. Right, we had this exact conversation this morning, in that we don't have control data, I guess, or a good ground truth data set that combines multi-omic data, and that at least would seem to be an initial starting point to, and now I'm getting into the solutions, but not having, how might we generate that ground truth data set that whereby some of these methods can be, you know, explored and shared. There's a lot of different diverse data sets out there. It's Yong. It sounds like you guys have a lot of experience on the brain cell census, on the different types of analyses that are being used.
Yeah. Yeah.
So what are any thoughts there on sort of the problems that you've had?
So it actually turned out pretty well on all the molecular data modalities so far used for the brain initiative cell census that can be integrated pretty well, aligned, so they can be aligned pretty well. So
do you use, can I just, so I can understand more, because it's not my field, but when you say align well, how do you know? What's your kind of reference point for that?
Yeah, so the data, so they generated about you know, from the same brain region. So we basically start from one common brain region, motor cortex, and so which probably has about 1 to 2 million cells, and then so all different groups use the same brain region sample to do their own experiment. And then after they get their own data, and then basically, you know, registers to the reference genome. And then in that way, you can basically align well different modalities.
I see. So you've got like common reference genes that you're all looking for?
Yeah, so this is a sequencing-based approach. Yeah, what Ananda was just talking about, you know, align between imaging and the sequencing data. So far, we haven't achieved that yet. But potentially with Murphy's type of experiment, so we can, so Murphy's with single cell RNA-seq data can be aligned pretty well. So in terms of cell types. So, you know, each corresponding cell types can be aligned pretty well. And then from Murphy's imaging data, you can then map with potentially other modalities. But we haven't done that yet. And so, for example, the immunohistochemistry, like gene-based approaches. And so if we can, you know, all map to the same coordinate framework, so the spatial coordinate framework, so at least at the region level, these data can be aligned together. Maybe not at a single cell level, but maybe at the brain region level, protein data, and potentially also other imaging data like calcium imaging or voltage imaging data can be mapped together with transcriptome data or proteome data, immunoimaging data. So that's the hope, but we haven't done that yet.
Do you think that, I mean, again, I'm not familiar with this level of multimodal analysis, but do you think the field needs guidelines yet, or are you there yet? Is it, because again, that was another topic that came up this morning in terms of multiplex imaging, combining in situ hybridization methods and with protein and common protocols and so forth. There seems, you know, not yet a lot of information out there on sort of workflows, and that's maybe where the value of reference data sets would come in. So at least from a sort of an initial training and then sharing best practices.
I think that's extremely important right now. I think all programs face that, right? We have that in the tumor atlas. And I mean, I guess it's both probably equally important. It's also important to advance using different modalities in the same sample. And I do think there are different questions, right? It probably comes down to what, first of all, what that group that you attended this morning, if they've already tried to tackle this, maybe it will be redundant. And second, what is this group most interested in, right? Because I think they're too diverse to tackle them both, right? I don't know.
Well, at least for this morning, we were more concerned with the lab side of things, the actual physical, the preservation, and the handling of the sample, and then obviously data downstream is a function of the quality of the sample. But so I think they're two different topics, but they're definitely intersected.
So one question I have actually is about what's really the advantage of to do, we talk about a lot this morning about multimodal analysis. So basically it's a simultaneous detection of RNA and ATAC-seq, for example, or protein and RNA. So what's the advantage of that on comparison with if you can do post-experimental joint analysis? That's not very clear to me.
Well, I think you're moving from correlative computational extrapolation to, kind of a definitive answer, right? If you correlate two data sets, it's probably,
Yeah, theoretically maybe, but right now, joint multimodal analysis, they typically got very sparse readouts. So like RNA ATAC-seq, so both are pretty sparse, even though this is from the single cell. And if you do from the single cell, single modality, you probably can do much higher detection, more genes detected. And so that's actually my question to people if they want to propose multimodal single cell multimodal analysis. So I want to basically ask them the question. So what additional information you will get that might be missed from the respective single modality assays?
So, Yong, might it be that when you are analyzing one single cell and you get both kinds of data from that cell, it gives you at least the state of that cell in that time, such that your data, as Philip was saying, that this is not just more correlative, it's more definitive, as opposed to if you do these experiments separately in two different labs, even though they are apparently from the same samples, you are not 100% sure they're exactly the identical cells. And so you don't know whether exactly the identical data you're getting at that time and the state of the cell.
Yeah, I agree with you in principle, yes. Yeah.
So what are the, I think it's almost time, right? What would be the questions to?
Okay, where do we find multiplex data sets?
That's a difficult question.
Well, don't you have them from the programs on the brain, and is that they do not exist?
We do have some. SNARE-seq from UCSD group. They have generated one.
You're talking about multimodal, right? You're talking.
That's a multimodal.
Yes. Yeah. They're much less common. I agree. I don't know how many we have.
So this is from my program, and so SNARE-seq so far, that's the one we have, and probably the other just published Nature Methods, quite a few papers published, but not big data sets. But you know, since they are published, they should be somewhere, right?
Principle.
I mean, is it across like tissue or just in vitro cell lines or things like that?
So for the brain, it's a tissue, from tissue.
Yeah.
So let's just assume we have all the data we need. What would be the point then, to compare methods, or what would the goal be? Given the ask for this group.
Yeah.
Um,
So we need to talk about this later, about moving forward, what kind of collaborative effort we can have across the consortium.
So what kind of information should these contain to be useful? Is that meaning, what is the context there? Is it the metadata, patient level, the clinical experiment? I guess all of the above.
Yeah. So all the metadata, I think, should be there, right? And some kind of a standard. I don't know how to develop some standard across different consortium. So different types.
Is there a standard? Is that a problem? Is there no standard today?
For the multiplex single cell recording, I'm not sure. There are some, not so many studies yet.
Okay, so maybe that's another, how might we do that?
Is it possible to generate some kind of a standard data set? Because I think that might help in terms of the method development. Some kind of a way because we need some kind of a really nice data that really has all the properties that requires the methods to be optimized. I think that is, without that, no matter which methods we develop, it will be difficult to assess them, any sensitivity.
Agreed. I mean, I guess that's a goal of all of the different consortia, but perhaps it's not, and again, jumping into the next session, but that seems to be a good collaborative effort to make sure those high quality data sets are available and that they're well annotated and that there's a space for, sort of common set of goals around those for validation across the groups.
But who's best to establish and derive that, though, right? I mean, I guess I have two questions. What is more, to me, it seems more challenging to integrate different data sets from different sources than to integrate multimodal sets from the same cell, although I'm sure that's a challenge. But I think for the multimodal, the challenge is really what we discussed this morning, generating the data, much more than analyzing the data. And since this session is focused on analysis, I feel the challenge that the field currently faces is how do we integrate modalities that are derived, maybe from the same sample through different, from different sections, from different approaches.
You're right, but do we have that much data out still with this context, within this context?
Yes, that context we will, I mean, H10 is putting those data forth. Well, now everything has been delayed, of course, but this year, HubMap has those data available. I mean, data sets, right? Say, you know, colon cancer, single cell RNA-seq imaging, all derived separately, and those need to be put in context. And I think that's, I mean, people are working on that. Obviously, I don't have the experience. I'm just curious if anyone on this call does, and what, you know, what's the best way to go forward and tackle that.
I think from the experience, from what I've seen, methods out there, they are still quite simple because they still are based on this manifold dependency, which means that it's really, they're integrating it, but they're making a very strong assumption on the underlying relationship between the data sets. And some commonly, I don't know if that is the optimal method, but I think that is itself, it's worth talking about, if that's the right way.
Mm-hmm.
But I think the additional issue is back to the question which we brought up earlier, is like, how do you validate? Yeah, so because you're integrating different data sets from different tissues. Let's say you do the methods, at the end of the day, how do you validate that part? And I think that is where we still have, and I think it's challenging.
Mm-hmm.
Because it's not just the method, it's like, how do you validate them? Assuming that they came from different tissues.
Mm-hmm.
Yeah, so I think from the Brain Initiative experience, so we actually initially get all these kind of different methodologies and different modalities, assay. And so there's a concern about how we can integrate after the data be generated. So it turned out actually for the molecular data sets, so at least I think from the joint analysis and publication, and looks like these data can be aligned pretty well based on different analytical approaches. So, you know, you can do like Liger published by Evan McCuskell's lab, and also the other single cell fusion from Evan Mukamel lab. So both actually reach the same cell types. So they are very closely, you know, comparable, and even though from different computational methods. So, so this is for the sequencing-based approaches, and I'm not sure for the other imaging spatial methods. This kind of modality, how to align imaging data with sequencing data. So this is not very easy. So far, I don't think. Probably it's a challenge right now. Yeah.
So how are we doing in terms of time?
You mean kinetic?
No. Right now, in the session time.
Oh, session time. Is it?
I think we're up to the end of this session, right? This morning they just cut us off, so maybe they're leaving us go this time.
No, this session that was done for Dr. Collins's talk. So this they said that they're not going to do it. It's up on us.
I got a through Google. It says it's now break time.
It's a 10 It's a 250, right?
Yeah, I think that, well, my Google says it's now break time back into, I can't read this.
15 minutes.
Yeah.
So I guess we can break when we want. I don't know if we have we defined a question for the next session? And how might we question? Which of those is the most promising for us to tackle?
Yeah.
Well, the reference data set seems like one important one to the potential follow-up. Control data. They're the two that stand out for me.